My name is Queenie.
My name's Haley.
Hi, I'm Dahlia.
And I'm Emily, and today we're going to be talking about Newcastle disease virus and how it induces apoptosis and inhibits migration in oral cancer cells. To start off, I'm going to be introducing the disease itself. So, the disease of interest is Newcastle disease virus, and it is a highly pathogenic viral disease that primarily affects avian species. The key characteristic of interest is its ability to affect cancer cells without affecting healthy cells. The two gene products of interest are beta-catenin and MMP7. Beta-catenin is a protein that helps in adhesion, proliferation, and invasion, and it is a central part of the transcription of oncogenes like MMP7. MMP7 is the other major gene product of interest, and it stands for matrix metalloproteinase 7. MMP7 is an enzyme that helps break down the extracellular matrix, and therefore it helps aid in promoting cancer cell migration and metastasis. To start talking about the actual paper, the paper sought to solve a main question, and that was, how does NDV inhibit migration and induce apoptosis in cancer cells? The specific hypothesis was that NDV disrupts the Wnt/beta-catenin signaling pathway and downregulates MMP to inhibit migration and induce apoptosis in squamous carcinoma cells. The diseased phenotype of interest is cancerous cells, but specifically the high levels of MMP and beta-catenin associated with metastasis and tumor progression. NDV modifies these pathways and reduces cancer cell viability and migration. The molecular mechanism leading to the high levels of MPP or MMP7 and beta-catenin are the increased activation of the Wnt/beta-catenin signaling pathway, because it causes the overexpression of oncogenes like MMP7, cyclin D1, and CMYC. NDV helps downregulate PAKT and PGSK3 beta. This leads to the degradation of beta-catenin and a decrease in MMP7 expression, which slows migration and causes cell death.
Okay, so now let's go ahead and discuss the roadmap of the study. So the general goal of the study was to evaluate the therapeutic potential of Newcastle disease virus as a treatment for human oral cancer. Specifically, the researchers wanted to determine how NDV induces cancer cell death or apoptosis and how it inhibits migration, which is both critical in cancer progression. The experiments found in the study focused on the roles of beta-catenin, a key signaling molecule, and MMP7, an enzyme involved in tumor invasiveness, to understand the mechanisms driving these effects. Since cancer cells exploit the Wnt/beta-catenin signaling pathway, beta-catenin, along with genes like cyclin D1, CMYC, and NMP7 levels are high, which promote tumor invasiveness. However, researchers wanted to experiment to see how infecting cancer cells with NDV counteracts this, finding that it actually downregulates PAKT and PGSK3 beta, and that promotes the degradation of beta-catenin and reduces MMP7 expression. So as a result, authors found that when the cells were infected with NDV, cancer cell migration was inhibited and cell death is triggered, which slowed tumor growth. Overall, the trajectory of this experiment wanted to establish how NDV was a promising tool for NDV and giving us insight for molecular anti-cancer action.
All right, so we are discussing the most establishing experiment now. It was a multi-figure experiment, but we chose figure 5C, where the authors were looking to see how beta-catenin levels were affected when the cancer cells were infected with NDV. And it also kind of served as a basis for further exploration of its role in NDV-mediated migration inhibition. So they performed 6 different Western blots for beta-cat, and then its beta-cat regulated genes, as well as NDV and beta-actinin. The results of all these Western blots showed that NDV, its protein, is expressed in cancerous cell lines when it's infected, and beta-cat is reduced beyond the controls levels as a result. And then beta-catenin targeted genes is also downregulated. There was 2 controls that were used. First, was the mock, which was just cancer cells that were infected with like a mock infection, like a mock vector. And then beta-actin acted as a loading control. And the authors concluded based off of these Western blots that NDV does in fact inhibit the transcriptional activity of beta-catenin. And yes, the category that we decided for this specific figure was expression and establishing. So moving on to why this was an establishing experiment, there was many Western blots, but the specific Western blot that focused on NDV protein expression was the most important establishing one. That Western blot not only showed that NDV is expressed at the protein level, it also provided baseline protein expression of NDV in the cancer cell lines. And then it also is the first experiment where the authors directly measured beta-catenin levels. The previous experiments measured beta-catenin through its beta-catenin regulated genes. So this experiment is independent to any of the prior indirect observations made from like measuring the fold activity of its target genes. So in this way, it generates baseline data for the future experiments that they did. And in the later experiments, after they confirmed that there was a relationship between NDV and beta-catenin, they tested how beta-catenin affects expression of GSK3 beta, which is a beta-cat regulator, and then also phosphorylated AKT and GSK3 beta, which are both GSK3 beta regulators. And then they also tested to see if NDV affected localization of beta-catenin in the cytoplasm and in the nucleus of the cancer cells.
So for our most important hypothesis-driven experiment, we believe that it is figure 5A from the article, in which it tests the question, in the presence of NDV, how is beta-catenin transcriptional activity affected? To test the experiment, they used top flash or FOP flash luciferase assay, in which they used the controls of mock cells, which are not infected with NDV, FOP particles, which contain luciferase, and lithium chloride, in which all cells are treated with the compound. The result that came from the experiment was that mock cells treated only with lithium chloride had an increase of luciferase activity by 41-fold, whereas SAS cell lines treated with the virus and lithium chloride had a decrease in luciferase activity. So from the experiment, the researchers concluded that in the presence of the virus, beta-catenin activity decreases, and the experiment itself is a functional experiment. So why is this considered a hypothesis-driven experiment? The experiment determines if the virus affects the expression of beta-catenin, and the experiment itself does not further test or confirm a previously made statement in regards to NDV affecting beta-catenin activity. This experiment applies to the overall hypothesis of the study, since beta-catenin upregulates the enzyme MMP7, which has been found to be upregulated in cancer cells in their migration in previous experiments made within the study. This means that if beta-catenin transcription activity increases, then the production of MMP7 also increases, leading to higher migration rates of cancer cells. On the other hand, if beta-catenin transcriptional activity is decreased, then the production of MMP7 decreases, which then leads to lower migration rates of the cancer cells. Since the virus NDV has been found to decrease beta-catenin transcriptional activity, then the production of MMP7 decreases, inhibiting the migration of cancerous cells.
All right, so to retest the findings of Figure 5a, where beta-catenin transcriptional activity was assessed using TOPFlash/FOBFlash luciferase reporter assay, an alternative method that we could use is chromatin immunoprecipitation followed by PCR. And we can use that to directly measure beta-catenin's binding to target gene promoters. The aim is to validate the effect of NDV infection on beta-catenin transcriptional activity in the SAS cells by assessing its binding to the promoters of its downstream target gene. And why is it a good alternative? The approach complements the original luciferase-based assay by directly interrogating protein DNA interactions. And then some advantages of this validation experiment include ensuring that being able to measure physical binding of beta-catenin to DNA, which provides more direct evidence of transcriptional regulation, and also the validation of downstream effects. We could see the links observed. We could see that the observed transcriptional activity is linked to the actual promoter occupancy.
And then for the different types of treatments and controls, we're basically looking to test for beta-catenin binding in cells that were infected with a mock vector, and then cells that were infected with NDV. The mock treatment serves as a negative control, which provides baseline information about beta-catenin under normal conditions. And then we also use a non-specific IgG control, which was used to demonstrate that only beta-catenin specific antibodies can pull down without viral interference. And then some positive controls used was the cancer cells infected with the strain, which was used to provide a visual for beta-catenin interactions with the target genes under the influence of the infection itself. And then also we looked at different target antibodies and how they binded to their target genes. And this was used to confirm that beta-catenin actually does bind to its specific target genes.
So for the experimental steps, the SAS cells will be grown and treated with either the virus NDV or will be in mock conditions. Then the cells will be cross-linked with formaldehyde to preserve the protein DNA interactions, and the chromatin will be sheared and immunoprecipitated using an anti-beta-catenin antibody. Then we will then go through PCR analysis in which antibodies will be designed to bind onto target gene promoter regions, such as cyclin D1 and CMYC, as well as MMP7. And the amount of antibody binding will reveal the level of beta-catenin's binding activity under different treatments. And to analyze the data, the relative band intensity of the chip samples will be normalized to input chromatin and compared across treatment groups.
Following Queenie's explanation, we can start to analyze the figure itself. So in the figure, we see that the mock cells have the baseline band expression, and the NDV's cells show that there's decreased expression in the beta-catenin, CMYC, and cyclin D1. This indicates reduced beta-catenin binding when compared to controls, and that indicates as a whole that there's a reduction in transcriptional activity in oncogenes. This is consistent with what was originally found in the original figure 5a. To read a little bit further deeper into that, though, this also means that NDV inhibits the Wnt/beta-catenin pathway, limiting metastasis.